Professor, tell us a little bit about your department and the scope of your research.
Okay, so I work at the Centre for Eye Research Australia, which is housed in the Royal Victorian Eye and Ear Hospital in Melbourne. So this is a tertiary hospital. It acquires patients from the whole of the southeast of Australia. So it has a very wide spread of patient samples. Within the centre itself, we cover everything from public health, genetics, epidemiology, World Health Organization Centre, through to stem cells, pharmacogenetics, pharmacology, clinical trials, basically everything you could imagine.
In your particular area of interest and expertise, tell us about that.
Okay, so I'm head of the Ocular Genetics Laboratory, so I'm very interested in genetic basis of disease. One of my main diseases that I work on is macular degeneration, and I'm very interested in working out how we can use genetics and other risk factors to help patients and sort of bring changes from the bench back to the patient population and improve their health, so that they can actually maintain their vision for as long as possible, or to stop them losing vision in the first place. And so that's the whole thrust of my research.
Turning to the field of ocular genetics, we know that AMD is a polygenetic disease. There are no monogenetic forms of AMD that we know of.
Mm-hmm.
What is your paradigm for looking at this disease and teasing out which of these gene and genetic factors may be most important?
Okay, so it's actually been a very interesting journey, and when you look back probably 20 years ago, AMD was thought of as just being an environmental factor causing it. There was no real genetic risk that was known, and it wasn't until the early 90s when people started to look at twins and families that they noticed that AMD seemed to run in families a little bit more often than it should do. So that gave us the first hints that there may be some kind of genetic involvement in AMD. And then the first genome-wide association study, where we could actually look at a number of cases with disease and a number of people without the disease and compare the genes or the genetic variants across each of those patients and look for specific regions which might influence disease. And in fact, the first GWAS, or Genome-Wide Association Study, was done in AMD. And NEI and the rest of the world is very proud of this fact. And it was done just on 100 cases and 100 controls, so a very small number compared to what we do today, where we've now moved on to the International AMD Gene Consortium, which has 70,000 patients, cases and controls. So we've actually moved from just a tiny number of people to looking at huge populations across the whole globe, literally. And that's been the most profound change that's happened probably in the last 10 years. So this has given us a great angle, a greater understanding of the genetic components of disease, and so we've learned a lot. On the flip side of that, as you mentioned, it's polygenic, but it's also complex, which means that it also has environmental factors. And so we've actually done a lot of work on environmental factors as well, which might influence AMD. And some of our studies have certainly implicated diet as being certain components in diet, like red meat or alcohol, can increase your risk, and other components like green leafy vegetables can decrease your risk. But these go hand in hand with genetic components. So it's not just a one-size-fits-all, it's a combination of different things.
If we look at the genetic factors, which of these genetic factors appear to be coding for or expressing proteins or other factors that might be involved in the pathogenic pathway?
So most of the variants or the genetic factors themselves tend to lie outside genes, which makes it quite difficult to work out exactly what they're doing. There's very few that reside in the middle of a gene. And this is typical for most complex diseases. So it's not that's why it's difficult. And it's not an easy thing to tease this apart, but certainly we're trying to look at ways and opportunities to get into these variants, see what they're actually doing, why they cause disease. And there's a number of experiments, not just in my laboratory, but all around the world that obviously people are doing this. And we're interested in what's used to be called junk DNA, which is all the bits in between the genes, the coding bits of the genes, which is the vast majority of each of our genome, which on the face of it appear to do nothing, but now we know that it's actually full of lots of things called regulatory elements, and a lot of these variants tend to fall inside some of these regulatory elements. So now it's trying to work out what these regulatory elements do in terms of activating or deactivating or changing the expression of a particular gene.
There's been a lot of controversy in this country with regard to recommendations for genetic testing for AMD. What is your position on that?
We did a study a few years ago trying to look at the available variants which were out there then. The list has obviously increased since then. And I understand the argument made by the Academy of Ophthalmologists, which is very understandable. And at the moment, we are in a position where we can't offer a genetic test. We can offer a certain risk, but that's about it. And really, we have a situation where we have too many false positives in the population if we try and screen in a population level. So this is not to say that this kind of testing is not useful. It might be very useful in high-risk populations, such as families, or people where there's much more increased risk. So if we can target those people, if we can arrive at a better and a bigger data set, then I think we're on the way to something great. We're not there yet, but we've certainly on the first footsteps to that process. So what we're trying to do in my lab is work at better prediction models rather than just basing this on just a small subset of genes. We might be bringing in different environmental factors. We might be looking at some clinical factors as well. And we're trying to work out mathematical models which will better predict who will go on to get disease and who will not get disease. So if we can do that, then that's another step forward towards offering a kind of a diagnostic or a predictive test to people who might get AMD in the future.
How far are you in the development of this algorithm?
So we've started. We have a lot of the components already in place. We've got some bioinformaticians who are interested in doing this, and now we're just trying to work out which components and which particular model we will put together to give us the best kind of prediction. Because there's many different mathematical models, there's many different factors you can put in, and it's trying to work out which combination is going to be the best one. Now, that's not to say that if it works in our population, it'll work in every population. It might not. There might be little tweaks, twists and turns that we have to do depending on what's in that population, because there will be certain cultural, there'll be certain risk factors that might be unique to that particular population as well. So we think we're going to get there in the next couple of years, or certainly we'll be working on this test anyway.
Turning to animal models of AMD, as you know, there are a variety of animal models that are relatively narrow in scope. Are there any animal models that are particularly amenable to genetics?
Animal models in AMD, as you mentioned, are limited, and the main problem with a mouse model is there's no macula. So that's a major drawback. So there's no one animal model that fits all, but certainly in the animal models that are commonly used, you're looking really at a defined feature, whether it's a thickened Bruch's membrane or an increase in lipid deposition or something, a specific change, rather than a one fit for all findings in AMD. So we are thinking that it would actually be better to use stem cells, and we're trying to work on growing, or we have actually grown RPE cells at our institute, where we've taken immunopluripotent stem cells and actually grown them into retinal pigment epithelial cells. And so we're attempting to try and use these to mimic the disease process. And so we'll be able to actually use those cells to maybe do drug targeting, or actually look at different factors, even genetic background, or changing the genetic background of those cells to modify disease and see what actually happens with these cells.
As you know, there's been a revolution in the last couple of years in terms of our ability to edit genes, the CRISPR-Cas revolution. And how do you see that playing into your paradigm of more accurate genetic quantification, so to speak, to getting an understanding?
Yeah, CRISPR is fantastic if you're doing a single hit, but it's advancing very, very rapidly. There are technologies which are now out there where you can get multiple hits, but of course there's the off-target issue of will you get the hit in exactly the right place at exactly the right time without getting other hits that you don't particularly want. And so that's the main issue at the moment, and certainly we're hearing a lot of advances, very rapid advances actually, where people are trying to minimize these off-target effects. So again, I think in the next year or two we're going to end up with a situation where people will be more satisfied with the technology. They'll be pushing ahead, and I think in like a stem cell model, a regenerative model, where it's in a petri dish, then it might not be such a big issue at the moment. Obviously, if you were going to put that particular technology into a patient, then yes, it's a big issue. But in a petri dish, I think we can actually move ahead now, and hope that off targets are minimised in those stem cells, and we can actually do some modelling now, and check what's happening.
Tell us about how you'd like to see this particular field evolving in terms of translational potential for patients.
So I think in the future, the ideal situation is that a patient would walk into the clinic, the patient would sit down, you'd take a sample, either a skin sample, or you'd take some cells from the eye, and you'd grow them up, and you'd offer some kind of CRISPR-type technology. You'd correct that defect or multiple defects, or you change the genetic landscape of that particular cell. You'd grow up the cell, and you'd implant that back into the patient's retina. Now, the issue there is, will you actually correct the change long term? How stable is that particular technology? And obviously, once you've taken those cells, then it will be hard to actually go back and improve vision. So I think you have to say, well, this is a line in the sand for this particular patient where we've actually done something at this point, and now we will preserve your vision so it won't deteriorate, but we won't be able to improve it. So again, we need to go back to understanding the basis of disease and picking up people very early on. So it's not at the late stage, that's too late, because people have already lost their vision. The patients are saying, I can't see. We want to go backwards. And so that's what we're trying to do. We're trying to work back to the early stages of disease, pick up changes that are happening in early vision loss, and saying, okay, we know that you're going to get worse, or we know that you're not going to get worse. And so therefore we'll come in with another technology, which could be some kind of pharmacogenetic basis, again, which we're working on. And so this could be where we recommend early treatment with some kind of anti-VEGF, or it could be some other adjunct technology that we add into that. So in terms of pharmacogenetics, that's exactly what we're doing now. We're trying to investigate and identify new genes that might be involved in response to treatment. And we've got some interesting candidates just at the moment.
Talking about early detection and the immunology of macular degeneration, are there specific biomarkers that you think are important or that you'd like to expand the use of?
Again, we have a study. In fact, it's been looked at in terms of blood, and we're looking at a series of different markers to try and work out whether there are certain biomarkers in the blood which could be used as a diagnostic. And we're also looking at different biomarkers which could be clinical parameters which might indicate that you are on the route to advanced AMD or progressing AMD. And so it will be a combination of factors. It probably won't be one single blood test or one single image in the eye. It'll be a combination of different factors, and that's what I imagine would be happening in the future.
Listening to this remarkable work that you're doing, what are the implications for them in the next years ahead?
So I think AMD is a world leader in terms of research, and I think the current patients, there's probably not a lot we can actually do for them in terms of improving their vision. We can certainly stop them progressing with the drug treatments that we have, which has been a miracle change. In the future, there will be lots of other drugs that come on the scene, which will be able to treat not only the neovascular or the wet form of AMD, but also the dry form. And so we're putting a lot of effort into actually identifying what determines this difference between the wet and the dry form. And we're doing some current work at the moment where we're looking at analysing the whole genome of a patient, and we're looking for gene-gene interactions between different regions of the genes to work out whether they're different between the two different end-stage types of AMD. And we have some early results already which seem to indicate there are distinct interactions in each of these subtypes.
Professor, thank you so much for a most illuminating, educational and edifying elucidation of what's going on. And we look forward to following your research and learning more about the results as they come forward. Thank you again.
Thank you.